Chapter 4, we'll take a tour of the cell, look at different types of cells, and their defining characteristics. The cell theory is composed of three components. The first being that cells are the smallest unit of life. You can't get anything smaller than a cell and still be able to do all those living characteristics. Second, all living things are composed of cells, meaning if you're alive, you must be made of cells. And lastly, cells come from other cells. They divide and give rise to the next generation through either mitosis or meiosis. But most cells are microscopic. They don't get very large. There are a few exceptions, such as our neurons, which are extremely long, but generally speaking, you must use a microscope to view most cells. We recognize that life is incredibly diverse, but we're all made of cells, and cells have some general principles in common. Three of those, what we call the three S's, are survival, sex or reproduction, and size, speaking about surface area and volume ratios. The surface area to volume ratio is really a ratio of the amount of membrane covering the outside of the cell to the internal volume, all the collective things inside. Surface area is calculated by a height times width, or a squared value. You then add up the number of sides to get the total number of surface area. The volume is calculated height times width times length, so therefore it's a cubed value. As a cell gets bigger and bigger, these two, surface area and volumes, don't grow at the same rate. The cubed value grows more quickly. And what that means is our cells have a finite size. They can't get too large. If you get too large, your volume internally is not matched with the covering on the outside to maximize exchange rates. What do we mean by exchange rates? Well, things need to get into the cell. Nutrients, gases, and things need to get out of the cell, products and waste. And if you don't have enough surface area to get that exchange rate, this cell will literally die. This is why, as humans, we're made of 6 trillion cells rather than one giant cell. So surface area to volume ratio makes it so most cells are within a given range, typically 1 to 10 micrometers. But all cells share other characteristics, such as being covered by a plasma membrane, being filled with cytoplasm, and then some other features, well, they all have genetic information, typically in the form of DNA in what is known as a chromosome. Lastly, all cells have ribosomes, and ribosomes are extremely important because it's where we make proteins. Recall the two different types of cells from the first week that we started the class. Prokaryotic cells are all of the bacterial cells. They exist in the domains bacteria and archaea. These cells are small and simple, generally 1/10 to 1/100th the size of our own cells. Internally, they only have ribosomes and genetic information all floating within the cytoplasm. Their genetic information is made of a single circular DNA that is located in what is known as the nucleoid. This is just a region, not an actual organelle. Prokaryotic cells are generally unicellular or colonial and can have cell walls that exist outside of their cell membranes. Please remember that all cells have cell membranes. Not all cells have cell walls. Bacteria make different types of cell walls, and that chemical composition can be useful in identifying different bacteria. An example of one cell wall is one called peptidoglycan. It's a fairly common one. Other structures can be found on the surface, such as pili and flagella. These are often useful in attachment and locomotion. Lastly, prokaryotic cells can make capsules. This enables them to hide, say if they're in our bodies and trying to avoid detection from our immune system, or even to hibernate. Think about food poisoning and those bacterial cells that are trying to hide and not be killed. Bacteria also come in different shapes, which help us to name and recognize them. You'll notice here several different names that often show up in the actual Latin name of the group. In one called Staphylococcus, this is one that gives us staph infections, each of the bacteria are cocci, which means round or circular. They can also exist as vibrio, which look like little commas or boomerangs, bacillus, which are little rod shapes, and even spirillum, which are spiral shaped. Eukaryotic cells are vastly larger and more complex than the bacterial cells. These include animals, plants, fungi, and the protozoans known as protists. Recall that we are 10 to 100 times bigger than the bacterial cells, and that means inside we have a lot more organelles. These organelles, many of them at least, are made of membranes, and each of those membranes gives them their unique features. The other difference is our genetic information. It's housed within the nucleus, and instead of being a circular strand, they are linear strands or fragments. There's a lot more diversity in eukaryotic cells, which is why we see them in many different kingdoms, including fungi, animalia, plantae, and the protozoans known as the protists. As we describe eukaryotic cells, we'll focus predominantly on animal cells, but we do want to present a little bit about plants. Both are similar. We have similar organelles that we share, such as mitochondria, Golgi, endoplasmic reticulums, peroxisomes, and the nucleus with our DNA. But plants have some unique organelles only to them, such as the chloroplast, which is where they do photosynthesis, the central vacuole, for storing water and balancing salts, a cell wall, and plasmodesmata. Those are channels between the cell walls. Animals, instead, have things like lysosomes and centrioles, and we'll talk about those in greater depth. Like bacteria, we have a surrounding plasma membrane. We're filled with cytoplasm, and there's fluid that the cell influences directly outside of itself, known as extracellular fluid. Now let's focus on the cell membrane. Recall from last week that the plasma membrane is made of phospholipids. This was a lipid category, meaning it was hydrophobic. Note the picture. We have a phosphate head, which gives it some polarity, a glycerol in the center, and two hydrophobic fatty acid chains. Our membranes are built of those phospholipids arranged in two layers, known as a bilayer. Note that the heads, the phosphate sides, point outward, and the fatty acid, hydrophobic tails, point inward. Together, those phospholipid bilayers create a selectively permeable membrane, or a membrane that's choosy about what can go in and out of the cell. That phospholipid bilayer has other structures floating and attached within those bilayers. First are proteins. A lot of our membranes have proteins, and they can either span the entire width of the membrane, or they could be just attached either externally or internally. These proteins are very important because they can often serve as attachment points, and those that span the entire membrane can serve as doorways for things to get in and out. We also have lots of glycoproteins and glycolipids that attach to the surface. Lastly, cholesterol is embedded with the fatty acid tails, and this gives a thickening and stabilizing effect to that membrane. Now that membrane gives several key things to our cells. One, that they're selectively permeable. They can choose what gets in and out, generally speaking. They can have cell-to-cell recognition, and that's what those glycoproteins on the surface are for. They also have cell receptors that allow communication between cells. Now, cell walls are only found in plants and fungi, never in our animal cells. Though they also have a plasma membrane, the cell wall sits outside or on top of that membrane. Cell walls make for more rigid protection and support to those cells. If you link them together with cellulose, you have the components of a plant cell wall. Fungi, on the other hand, make their cell walls out of chitin. In between cells, bridging the gap between the walls, are plasmodesmata, basically channels that allow cells to communicate and to send products back and forth quickly. Now let's look at animal organelles. In general, the term organelle means little organs, and that's because each of these structures within the cell is carrying out, well, a similar function to what your organs do in your body. Some are dedicated for energy and processing, some are building, detoxifying. Everybody has a role. Let's start with the nucleus. Now the nucleus, remember, contains our genetic information in the form of DNA. I mentioned that DNA are in linear strands, what you might think of as fragments, but that's in comparison to bacterial DNA, which is only one circular strand. Each species in the eukaryotic cell have a unique number of these DNA fragments or chromosomes. They're all loosely spread out inside of the nucleus in a form known as chromatin. The actual membrane outside the nucleus is known as the nuclear envelope. It is a double-layered structure of phospholipids that surrounds and provides a nuclear pore or a hole through which things can get in and out of the nucleus. Lastly, dark dots will often appear inside of the nucleus, and these are called nucleoli. That's plural for nucleolus. These are factories. This is where we produce things like RNA and the components to making ribosomes. So here's a real look at a nucleus inside of a cheek cell. Notice it just looks like a dark blue circle with little darker dots inside. So recall, the outer membrane of the nucleus is made of the nuclear envelope that has pores to allow things in and out of the nucleus. And notice you can't see the actual DNA because it is spread out and fills the entire nucleus in a form known as chromatin. You might be able to detect darker dots inside the nucleus. Recall, recall that the nucleoli inside the nucleus is where we build the components of ribosomes. Well, ribosomes are made of two components. They're made of proteins and RNA. Those come together to become functional organelles that work outside of the nucleus in the cytoplasm. Now, ribosomes are where we build proteins. So it is said that this is the site of protein synthesis. Ribosomes can be found floating in the cytoplasm, but in a great many of our cells, they're in higher density actually attached to another membrane called the endoplasmic reticulum. The two different components, or halves of a ribosome, called the large and small subunits, are just the top and lower portion of the brown image in the diagram. The next organelles we'll talk about are endoplasmic reticulum, and there are two types. Both are reticulum, or little network. That's referring to folded up phospholipid bilayers. So these membranes serve different functions because of, well, one, their appearance. The two types of endoplasmic reticulum are rough and smooth. Rough endoplasmic reticulum, often called rough ER or RER, have ribosomes attached to them, giving them the appearance of having the measles. Smooth are these tubular Recall that rough endoplasmic reticulum is rough because of the ribosomes attached to it. That means its function is really closely linked to the ribosomes. Together with the ribosome, it aids in building our proteins. Smooth endoplasmic reticulum are tubular in nature and serve as a great storage area. This is especially important in our muscles and nerve cells where calcium is extremely important and we need in high concentration. Smooth also plays a role in detoxifying components inside the cell and in lipid synthesis, especially the plasma membrane. Another organelle that is made of folded up phospholipid bilayers are called the Golgi complex. This system of folded membranes aids in receiving, modifying, and then repackaging and shipping out things like proteins. This is an important checkpoint. Often anything entering a cell must come to the Golgi first, and then it's decided where to ship it internally. I like to think of this as sort of a shipping hub, like UPS. Once those packages are shipped out, they're done so by being wrapped in a small membrane vesicle. These vesicles come in a variety of types, and we'll look at a couple of them. One are called peroxisomes. This is where fatty acids are packaged. In this diagram, you can see how this all shipping and receiving works. So let's follow a protein that's produced on the ribosome and packaged by the rough endoplasmic reticulum in what is known as a transport vesicle. This is just a little phospholipid ball. That vesicle arrives at the Golgi and fuses with it. Those proteins are released and moved through the Golgi. In there, they may be modified or even activated. The Golgi then repackages them in a secretory vesicle, which is shipped to the actual cell membrane or outer plasma membrane, where it's fused and those proteins are released. Think about your digestive tract producing digestive enzymes. This is how they're produced at the ribosome, yet work outside the cells in your stomach or small intestines. Now I mentioned the Golgi produces vesicles, and there's lots of different types of vesicles. In animal cells, lysosomes are a very important vesicle. This one contains digestive enzymes, but these enzymes work inside the cell, and they help us in digesting components and things that are brought in, or even neutralizing things that could be harmful, like bacteria and other pathogens. They also help in the recycling of old worn-out parts or organelles. If you haven't heard me say phospholipid bilayer about a dozen times yet, well, here's some more organelles made of phospholipid bilayers. Vacuoles are big, open type of organelles that often have fluid filling inside them. We discussed 3 different types. Food vacuoles, well, they store food. Think about adipose fat cells inside your body. Their food vacuoles are filled with fat. The central vacuole is a type of vacuole you see in plants, and this is where they store their salts and water for balancing what they need internally. The contractile vacuole is kind of a unique vacuole in that it has proteins that are able to contract and squeeze. The contractile vacuole is really for regulating water for cells that live in an environment where water literally is streaming into their cells, and if they don't deal with the extra, they'll explode. This is typically found in the protozoans that live in fresh water. Another organelle that has a phospholipid bilayer surrounding it is the mitochondria. This is known as the powerhouse because this is where we produce the greatest amount of cellular energy. That is, where we convert our food into ATP. Now this organelle is unique. One, it's got a double membrane barrier, so 2 layers of phospholipid bilayers surround it. The internal one folds inward into internal folds called cristae. Also inside are ribosomes and its own circular DNA. That DNA is very much more bacterial-like. The number of mitochondria in each of our cells can vary from hundreds to thousands. That depends on how energy demanding each cell is. Next is chloroplast. Now I realize I said I was focusing on animal and human cells, but I did want to bring up this unique organelle. This is found in plants and some of those photosynthetic protozoans. This is where sunlight is turned into carbohydrates. They contain the pigment chlorophyll that absorbs that solar energy and aids in the production of food. What's unique about the chloroplast, it is double membraned around the outside edges. It has its own circular DNA and its own ribosomes. Recall what other organelle I just mentioned that has similar features. So those unique features of mitochondria and chloroplasts have led to the development of a theory called the endosymbiotic theory, or endosymbiosis. We believe that hundreds of millions of years ago, a small prokaryotic bacteria cell was engulfed by a larger eukaryotic cell. Probably it was going to eat it, but for some reason it didn't consume it, and rather That bacteria cell lived internally, producing energy. The two began embarking on a relationship that was a win-win for each other. That is known as a symbiotic relationship. After millions of years, that symbiotic relationship has become a reliant one, and all eukaryotic cells have mitochondria. Chloroplasts, we believe, came about later on, but through a similar process. So, a eukaryotic cell engulfed a prokaryotic cell, incorporated it internally into its own set of organelles, and now a complete reliance has developed. We also see inside, in the cytoplasm and surrounding the organelles, a scaffolding of proteins known as the cytoskeleton. These help give it fill and rigidity, serve as anchor points, and even ways to move things through the cells. We generally look at three different types of protein fibers: microtubules, intermediate filaments, and microfilaments. Microtubules are essentially a whole bunch of protein molecules wrapped into an open tubular structure. These help in moving things through the cells, creating vesicle transports, as well as serving as anchor points in structures known as centrioles. Intermediate filaments fill up a great portion of the internal cytoskeleton. These help many of our cells that see a lot of stretching and distortion. It helps prevent them from exploding. helps them maintain their overall shape, and can anchor things. The last category are the microfilaments. These are long, linear protein chains that are best noted for their muscle contractions. Think of all the muscles that you have. They contract because microfilaments are pulling and shortening. They also aid in cell movement in single-celled organisms such as amoebas. Amoebas produce pseudopods or false feet using these microfilaments. Lastly, when we do cell division, when one cell divides into two, well, it's microfilaments that really help us in pinching off and creating two separate cells. Other structures that can be found externally are things like cilia and flagella. Cilia are hair-like cellular extensions. They're most common along our respiratory pathways, where the cilia actually filter the air and prevent harmful things from getting down in our lungs. Cilia are generally short and numerous, whereas flagella tend to be fewer in number and much longer. They're generally like a whip-like tail. In humans, they're only found in our sperm cells, but they can be found in other locations, such as in the protozoans. Now, bacteria have flagella as well as us, but the flagella inside of a bacteria has a unique microfilament arrangement that is different than a eukaryotic flagella, so they are actually species-specific. Around the outside of all of our cells is something called the extracellular matrix. Matrix is any structure or substance that directly surrounds a cell that is made and influenced by the cell itself. This is an area where support and communication between cells and even a protective barrier can be produced. Some cells produce collagen, such as in our skin, to help it give it that elasticity. In bones, it is the cells there that produce that mineralization of that hardened structure around them. Cells in multicellular organisms, such as animals and humans, have things called junctions. Basically, it's where the two cells connect or join to each other. We'll talk about three general categories, although there are others. There are tight junctions, anchoring junctions, and gap junctions that we'll address. Tight junctions are seen between cells where we need to make sure there's absolutely no leakage. In essence, a waterproof barrier. This would be really important in your stomach. Think about all that gastric juices, that pH of 2. We don't want it to leak between the cells, so tight junctions put those cells incredibly connected in such a way that nothing can sneak through. So you'll see this predominantly in our digestive tract and even our urinary tract. Now, anchoring junctions are basically protein microfilament rods that join from one cell to the next, much like an anchor going from one structure into another. They also go by the name desmosomes, and they're mechanically holding the two cells together. These would be important in cells that are being stretched or pulled apart, such as your skin or even your heart. And how about the uterus if a baby's growing inside? The last of the junctions is the gap junction. It'll go by other names, but I like the name gap because it's implying that there is an open channel between one cell and its neighbor. So a protein bridges between one membrane and the next and allows for communication from one cell to its neighbor in a very quick way. This is a pretty rare junction. In humans, we typically only see this in our heart cells. Heart muscle must contract uniformly together, and it's these gap junctions that allow us to communicate and coordinate all those cells to contract at nearly the same time. So let's recap what we've talked about. We described the cell theory. We talked about how surface area and volume ratios affect a cell. You were presented with different types of cells, prokaryotic and eukaryotic, and we looked at different types of organelles, their functions, and which types of cells contain them. These are all the topics that you'll see